Now, after discussing glycolysis, then the fate of acetyl coenzyme A and the fate of pyruvic acid. When pyruvic acid does get converted to acetyl coenzyme A, it is time for us to encounter the continuation of the aerobic respiration portion, which is the Krebs or the tricarboxylic acid cycle. So this one happens in the mitochondria, unlike in glycolysis, wherein the site of the metabolic pathway is in the cytosol. So this happens in the mitochondrion. And so it involves several steps, wherein the purpose is you combine stuff and then you release them in form of carbon dioxide, but in the process you gain cofactors which could be converted to ATP. Alright, so we're talking about the desire to produce NADH, the desire to produce FADH, anything that would produce ATP at the end, at the expense of removing carbon dioxide molecules along the way. So we will see CO2s, alright? We were supposed to see CO2s being released along the course of the tricarboxylic acid cycle. Alright? So the first step, alright, this is step number 1. I'll label them all just to get this over with. I don't have to write the step numbers later. So the first step is we have this 4-carbon dicarboxylic acid. The name of this one is oxaloacetate or oxaloacetic acid. If you haven't viewed my video on the introduction to the acids encountered in glycolysis and the Krebs cycle, I recommend you view it so you would be more familiar with the acids. So this is oxaloacetate, and this is of course acetyl coenzyme A. When you combine these two, so this goes in, you have 4 plus 2 carbons equals 6 carbons. And the process here is a combination or a condensation. And the enzyme responsible for this is known as citrate synthase. So from the name of the enzyme itself, we would be able to assume the name of the product, the 6-carbon acid, known as citric acid. So if you're wondering why the name is also known as a tricarboxylic acid cycle, or it's also called a citric acid cycle, right? Citric acid cycle. The tricarboxylic acid we are referring to here is citric acid because citric acid has 3 carboxyl groups. Let's count. 1 carboxyl group, 2 carboxyl groups, 3 carboxyl groups. So this is the tricarboxylic acid that we are talking about when we say tricarboxylic acid cycle. Lalang trivia. Anyway, now that we have produced citric acid, alright, out of oxaloacetate, so we have the 4 carbons here and the 2 carbons from acetyl coenzyme here, which manifested as these 2 in magenta. The next step would be conversion of citric acid into its, what happened, the OH at 3 became relocated at at carbon number 4. So there is only a rearrangement between positional isomerism or positional isomers, that is. So here we produce the isomer of isocitric acid, which is of acetic acid, which is isocitric acid. So reaction is simply isomerization. Now the enzyme here is named as aconitase, and you may be wondering, where is aconitase? Where did it get its name from? Well, actually, the conversion of citric acid to isocitric acid is a net reaction. Since there's an intermediate in the middle, citric acid is actually converted to cis-aconitate. And cis-aconitate is citric acid dehydrated. So how do we draw that? So COOH, CH2, COOH, CH2, COOH. What happened in the transformation of citric acid to the intermediate cis-aconitate is that you remove this and you remove one of the hydrogens. In exchange, what happened is that there is a formation of a double bond. Alright, so there is a dehydration reaction at the start. Then you rehydrate it. So you remove water, you add water. Sometimes chemistry makes itself repetitive and redundant. Anyway, so the moment that this one is rehydrated, instead of adding to the carbon number 3, it adds to the other carbon that holds a double bond in cis-aconitate, which is carbon number 4. And voila, the end reaction is simply isomerization. Alright. Now, the third step is conversion of isocitric acid into a 5-carbon compound. So what happened? There was a release of 1 carbon, and how is that released? In the form of carbon dioxide. So this is what I mentioned earlier. There is a release of carbon dioxide in the middle of things, and this is one of those steps that does this. So that particular reaction is known as decarboxylation. You remove a carboxyl group. Where, you may ask, well, the only point of difference here is that we have 1, 2, 3, 4, 5 carbons, 1, 2, 3, 4, 5 carbons, but here we have a carboxyl at 3. We have no carboxyl at 3. So it is this carboxyl at number 3 which is liberated as carbon dioxide after step number 3. Alright? And in addition, at carbon number 4, we used to have OH in isocitrate. But here, we have at carbon number 4, a carbonyl group. So this one was oxidized. So it's not only decarboxylation, but also oxidation. So this one is completely known as oxidative decarboxylation. And the enzyme responsible for that is isocitrate decarboxylase. Okay. So I think the enzyme itself is very self-explanatory. Although it also has another synonym, isocitrate dehydrogenase could actually apply, but dehydrogenase is the same as oxidation, you know. This could actually be helpful because isocitrate decarboxylase refers to decarboxylation, and isocitrate dehydrogenase, the synonym, refers to the oxidation process. So that might help you recall that there are 2 steps going on, or 2 reactions going on in step number 3. Okay. And in the formation or in the release of carbon dioxide, we have aided an NAD in becoming its reduced form. Alright. And here, in the next step, in step number 4, from 5 carbons, we go 4 carbons. So the same happens as with step number 3. You release a carbon in form of carbon dioxide. Now let's try to search where in this particular compound known as alpha ketoglutarate we will find the CO2, which will be released later on. COOH, the same. CH2, the same. CH2, the same. CO, the same. But there's a COOH here. There's no COOH here. So it is this carboxyl group which was erased or decarboxylated and then released as carbon dioxide in this particular step. So again, the reaction is the same as with the previous one. Oxidative decarboxylation. Alright. Oxidative decarboxylation. And the enzyme here is, owing from the name of the substrate, alpha ketoglutarate dehydrogenase. Very similar to the name of the enzyme isocitrate dehydrogenase. Alright. So, just like in this step wherein NADH is formed along with the release of carbon dioxide, here we also have the same reaction. So, along with the release of carbon dioxide, we produce another mole of reduced nicotinamide adenine dinucleotide. In order for us to form this 4-carbon acid known as succinic acid, but it has a coenzyme A, that's why it's named as succinyl CoA. Alright. And in the next step, step number 5, what happens is that simply the coenzyme A would be removed and everything else is the same. So from succinyl CoA minus CoA is simply succinic acid or succinate. It's the same. Alright, and the enzyme the thing that happened here is substrate level phosphorylation, and this is very weird, right? You may be asking, where is the phosphate here for you to say that the reaction is phosphorylation? But let me explain. The moment that the coenzyme A was released, phosphate came in but immediately the phosphate was taken up by guanosine diphosphate and was transferred to GTP. And the one that received the phosphate at the end is the guanosine nucleotide, because from the diphosphate it received the phosphate, it finally became a triphosphate, and this is something that we can convert later on to ATP. Very complex now, but at the end it still produces energy. Alright, and the enzyme responsible for this is succinate. No more space for the enzyme name. Succinate thiokinase. Alright, or another name of the enzyme is succinyl-CoA synthetase. And this is very deceiving because we assume that this enzyme facilitates the creation of succinyl-CoA out of succinic acid, but in the normal flow of the Kreb cycle, this enzyme does the opposite. Alright, does the opposite, meaning from the CoA with CoA to without CoA. Alright, so the reverse reaction is being done here. In the next step, succinic acid is now transformed into its unsaturated form. So one hydrogen for each of the carbons in the middle are sacrificed for the formation of a double bond. And the name of this unsaturated dicarboxylic acid is fumarate or fumaric acid. The enzyme responsible for the production of fumaric acid out of succinate is an oxidase, alright? Because technically, when you remove hydrogens, it's the same as oxidation, such much as reduction means addition of hydrogens. So this is succinate dehydrogenase, simply because you removed hydrogens, right? It's very it's very easy to understand. And simply the name of the reaction here is dehydrogenation, the removal of hydrogens. Okay, so here in the production of fumarate, there's a release this time of another reduced cofactor, but not anymore NAD, but here we are dealing with FAD converted to its reduced form. Alright, so this is the only time that we encountered this so far. Next, fumaric acid will then be transformed. What happened? The double bond was removed, and then you added water. So simply the reaction was hydration. You added water to the double bond, you remove the double bond, and you added OH to one of the carbons in the middle. Alright, the name of the product right here is malate or malic acid, and the enzyme responsible for this, alright, the name of the enzyme responsible, I can't find my pen, it's just here, my mind is processing, what color do I use? Alright, the enzyme is responsible for this is fumarase. Alright, so fumarase converts fumaric acid into malic acid by hydration. Okay, and finally, we have malic acid, and it is what happened to malic acid for it to become oxaloacetate. The OH became double bond O, so there was oxidation. Alright, there was oxidation in the hydroxyl group, and the enzyme responsible for that is known as malate dehydrogenase, which is almost similar to meaning malate oxidase. So that's what it did, oxidize the OH to double bond O. And in this particular sequence, we also have, even though there's no release of carbon dioxide, there is creation of NADH out of NAD. So out of the 8 steps in the Kreb cycle, there are 1, 2, 3, 4, 5 steps that are energy producing. And that's a big thing because when they are converted, that's a lot of ATP. But for the calculation of ATPs produced from Kreb cycle as well as glycolysis, that would refer to another video. But for now, we end the Kreb cycle here.